So good afternoon, ladies and gentlemen, and thank you so much whosoever is present here. And the reason for having this IC is that still now, if you look at India per se, India was the first country to have any kind of biosimilar anti-VEGF across the globe in 2015. And now we have come across, quite far, and we are in 2023, but still, if you really look at the biosimilar understanding, not only in India, means India should have even more, because we have been using biosimilars since last, means quite many years. Still, biosimilar understanding is not that great that we expected. So that's the reason that, in this next an hour or so, we would be trying to cover each and every aspect of biosimilar anti-VEGF with the help of our esteemed colleagues and teachers, who have agreed to be a panel on this particular program. So I would like to introduce each and every one. So first of all, Dr. Mangat Dogra, means he does not need any introduction. You all know him, means one of the pillar of retina in this country, because retina is whatever we do in this country, retina owes a lot to him. So right now he is in Chandigarh, and he would be sharing his experience in PGI Chandigarh, and what world can take cues from this country. That's what he's going to talk about. And then we have Dr. Debdulal. Dr. Debdulal works at one of the most busiest center of this country, in Disha Eye Hospital. And he has published a lot on the real world part and real world data on various biosimilars. A wonderful colleague to have him here. So he would be talking about his real world experience, because he is from very busy clinic. And then we have Dr. Tiwari. Means Dr. Tiwari's he deals with a very different kind of dynamics in Bihar. He works in Bihar, so he encounters a lot of financial, differentiation, a lot of different kind of patients. So he would be sharing his experience that, how financially it differs, and it has an implication as far as biosimilars are concerned. And then I have two of my gems right here, Nicola Parachuri and Nilesh. They were my students. But I was their student for many times, at many occasions. So a big thanks to them that we stand tall at this point across the globe as far as biosimilar area is concerned. Nicola Parachuri is a prolific vitreoretina surgeon at Shankara Eye Hospital, Coimbatore, and Nilesh is heading his Madhavi Netralaya at Ara, Bihar. So welcome each and everyone. And Dr. Jaishankar is there. He would have our keynote address. Please welcome him. He will be talking on retinal vein occlusion. So we have a slight change, because we wanted to start with Nilesh, but because Dr. Tiwari and Dr. Debdulal has another session, so we would start from them. So I would like to invite Dr. Debdulal first. He would be talking about the real world data on biosimilars. Dr. Debdulal, please.
Thank you so much, Dr. Ashish, my very good friend, and it's a pleasure to be here with him, and also Sir Mangat Dogra sir, our teacher, and my regards to him, and Sir, you too, and also Dr. Nilesh and Dr. Nicola and Dr. Satya Prakash, and I'm really thankful to be part of this team. And Dr. Ashish is someone that I always look forward to as far as anti-VEGFs go as a whole, and because he has his own long-standing association with anti-VEGFs, and we've learned, all of us have learned a lot on how to use anti-VEGFs. So basically I'll be speaking on our experience with biosimilars. So my disclosures. So basically we'll be dividing this into three parts: need for biosimilar anti-VEGFs, safety of biosimilar anti-VEGFs, and efficacy of biosimilar anti-VEGFs, and we'll talk about these as we go along. So today, there's absolutely no doubt that anti-VEGFs are necessary if you are to treat retinal vascular disease, and there's ample data that shows that there's been a lot of reduction of blindness, so to say, due to retinal vascular disease once the anti-VEGFs came along. However, treatment may be relatively expensive with biologics, and this can restrict patient access and treatment lengths, leading to suboptimal clinical outcomes. And this is important not only for the country, for our country and the developing world, but also in the developed world, where they are now increasingly looking into biosimilars. So part of the problem is a significant unmet need exists in many of the patients with retinal vascular diseases, one of them being unresolved fluid. So you need to go on and on with the injections. It's not a one-off game. And you have to have patients having to agree with repeated injections, because unless there are repeated injections that are possible, it's not possible to treat retinal vascular disease. But the problem is, if you have look at the luminous data, which is probably one of the largest data that we have on usage of anti-VEGFs. So if you can see here, half of these patients actually have maybe 3 or 4 injections at the most. And if you combine 5 injections, that then adds up to 60%. So, and this is something that's not good enough, because if you are to treat, say, for example, diabetic macular edema and, provide some gains to the patient, then you need to have somewhere between 5 and 7 injections, which is a rarity in our country actually with patients. And similar is the incidence or is the results with neovascular AMD. You need to have really at least maybe 5, 6 or more injections to actually have some tangible results. Now, in the Indian context, what is probably very, very important is non-affordability to the biologics which are there in the market. And this is where I compliment the biosimilars to start with, of course, the Intus company for providing something made in India for the Indians and also for the developing world and beyond. So, and at this point in time, it's very important to actually, talk about this important publication that has come up in the American Journal of Ophthalmology just a little while back by my friend, Dr. ASHISH and his group. And what he goes on to say in a nutshell is, biosimilars hold the potential to reduce the financial burden of highly efficacious biologic therapy in retinal pathologies. And this is something that's going to, get better with time. So what's holding us back from using biosimilars? Well, this is again something that he has spoken on, and in the eye there was this very important publication that came up. So this is because of the widely popular perception that biosimilars being generic versions of innovator molecules. I'm quoting Dr. ASHISH, and that brings a nocebo effect, that is negative perception amongst the care providers and the patients. So this is something that actually needs to be dealt with. This needs to be done away with. Now, to dispel the doubts, let's check on the safety and efficacy of biosimilar ranibizumab. Now, at this point in time, I should tell you that at Disha, our biosimilar experience is mostly confined to Razumab, and this is something that we've used for more than 25,000 procedures. And of course, as you know, Razumab has been used over the world for more than 5,000 procedures as of now. So let's talk about safety, because safety of a biosimilar is something that we look into right from the word go. And I'm happy to share this with you. We actually have the largest data as of date for biosimilars, as far as the safety of biosimilars goes. And this is 9406 Razumab procedures that were performed on 6404 eyes. And what we saw was that with Razumab, the adverse events seen were around 21%, but 97% of these were non-serious adverse events. And these adverse events were not very different from what was seen with the innovator molecules. Also, our large sample size detected a very minimal endophthalmitis rate of 0.01%, which is in keeping with other published literature from some of the better places. So to conclude, in a real-world setting, intravitreal Razumab therapy has an acceptable ocular and systemic safety profile in management of chorioretinal vascular disease. So this is our publication, so I'm going to beat my drum, but let's look at what others have to say. And the pioneer in the field, Dr. ASHISH Sharma, says that no alarming immunogenicity with Razumab was noted, and this is something that he has published in the eye. So, there's, hardly any doubt today amongst us users, and also beyond that, there's Razumab is something safe. The president of AIOS, Lalit Verma sir, our teacher, has also, published on this, and he has, shown that Razumab is safe and effective in the short term in DME, NAMD, and also RVOs. So we'll like to move on to the next, publication that we had. This is on DME, and if you notice here something very interesting, right from the word go, from month one, you have a statistically significant improvement in the vision as far as both innovator and biosimilar ranibizumab, Resumab here to be precise, goes, and this actually lasts out and right up to month 12, and there's hardly any difference between the two drugs, the innovator molecule and Resumab, or the biosimilar molecule. So all along the way, this is something that's working just like the innovator molecule. And as far as the central subfield thickness or the central macular thickness goes, again you can see there's a dip, and once the loading dose is done, there's a significant dip in the CMT values, and this is how it lasts out throughout the year if the patient is compliant.
Dibdulal, just to interrupt, sorry to interrupt, just go on the previous slide.
Yeah.
Sorry to interrupt you, but just keep this slide in my mind, because this is a very important slide. Although this is his real world data, but this is a very important slide from regulatory perspective. So if you really look at this 1 month and 2 months, that's where if you see, both the drugs, they converge. One is the innovator molecule and another is a biosimilar molecule. So the kind of effect that you see in 1 month and 2 months, that means at 4 weeks and 8 weeks. So that is a sweet spot for all the biosimilars when the primary endpoint is placed. So because always there is a question that, these trials are very short, primary endpoints are very short in these drugs, how do we believe them? But here, this is the reason, as he also rightly pointed out, that maximum effect that you see from these drugs comes around 4 weeks till 12 weeks. If you see, then there is a plateau. So you cannot really judge the equivalence if you have your endpoints later. So that's the reason, rather than, thinking that, because the trials were short. So just sorry to interrupt, but it was a beautiful slide.
I think this is something that's very, very important you pointed out. And I think, this is why I actually, ask him for his articles as they come out. Because, his is an experience, his is an bird's eye view of the whole scenario that he's been sharing with us for all this time, and this is something that's very important, and we learn each day with each publication that he comes out with. So regarding safety, and efficacy in DME, there's hardly anything to choose between the innovator molecule and Resumab or the biosimilar molecule. So moving on to the next publication that we had just a month back, this is the BALANCE trial. This is on neovascular AMD, comparing the innovator molecule of ranibizumab with biosimilar, that is Resumab here. And as you can see, again from this is the best corrected visual acuity. IRM stands for the innovator molecule, BRM for the biosimilar molecule, Resumab in this case. And as you can see, the visual acuity performance is pretty much similar, and right from month 3 after the loading dose, you get a substantial improvement in vision, which is statistically significant, and there's hardly any difference between both the visual acuity and the central subfield thickness in both these groups. There's an improvement at 3 months, which is just what he was pointing out for the DME article also. And this is something that lasts out, and this is something that you can drive with biosimilars, because the patients are more willing to kind of invest in this biosimilar, which is relatively less expensive. Now, intraretinal fluid and subretinal fluid, we keep talking about fluids in retinal vascular disease, and this is something that the patients had a similar response to with both the drugs. And more importantly, do you need more biosimilars than the innovator molecule when you treat a patient in these groups? Well, not really, because the numbers required were pretty much the same in both groups, and there was no statistically significant difference. And of course, the adverse events profile was pretty much similar. So intravitreal injection of innovator and biosimilar ranibizumab shows similar efficacy and safety in Indian eyes with NAMD. And I think it's time that all of us need to change and need to believe that, things produced in India can be as good as any in the world. Thank you so much.
Thank you so much, Dr. Dibdulal. And if anybody, Dr. Mangat, you have any comment on the real world data, what Dr. Dibdulal has presented?
I think Dr. Dibdulal has always given important direction, especially in using anti-VEGFs, you already said. And he has very well shown from the studies that I think I need not really add anything, because we have to have evidence, and he has shown that, and which is so important. That is what we need more and more from this country. So I must congratulate you for that. Thank you.
Thank you. Now, anybody has any question in terms of Debdulal sir's presentation? Then otherwise we'll discuss at the end. Now I would invite Dr. Tiwari to present his talk. Dr. Tiwari is going to talk about financial aspects. And once again, I think people were not here. So he practices in Bihar. So he deals with a lot of different kind of financial dynamics. So his talk would be very fruitful for the country like India.
Thank you so much, sir, for giving me opportunity in this instruction course. And I'm going to talk about financial implications of biosimilar. Finances are very important aspect of any drug. If a drug is costly, the uses are less, then we all know a phenomena in pharmacology known as orphan drug syndrome. It's just like if a drug is very costly and not used very commonly, there is no taker of that drug, and that drug generally gets out of market. Just we saw recently photodynamic therapy is no longer available, although many of its indications have gone into the backyard, but still we needed that treatment also. But due to financial things, that treatment is very less commonly available in our country. Biologics are very important, and their finances are much more important, because the development of biologics needs a huge budget, big finance, and then all those companies who invest so much on research and development, they want to take back that money once that treatment or that trial is successful. So the cost is very high, but that high cost impacts the success of treatment and the patients getting benefited from those biologics. Once that innovator biologics becomes off patent, and that is open for any development of biosimilar, then here comes the role of biosimilar. And why they are important? Because biologics imparts a huge financial burden on any country. Forget about a country like India or a poor socio-economic state like Bihar, where I am practicing. Even finances are very important for developed countries like US, UK, European countries. That makes a huge burden on even their financial system also, NHS or whatever, private insurance and all. So for the safety purpose, as we all heard from Debdulal sir's talk, that first and foremost important thing for the success of any biosimilar is the safety and efficacy. Safety is the primary success criteria, and once it is safer, then efficacy comes into the role. Once biosimilar is safe and that effective, then comes the role of the finances. If it is financially viable, we have from our experience come to know that it reduces the treatment cost around 50% for any kind of patient. And where we are using these biosimilars, unfortunately, the treatment repetition is very high, like in neovascular AMD or diabetic macular edema or IPCV kind of patients. We need to inject more and more injections. So that burden is very high. Older publications also show that on an average, at least one patient needs around 20 to 30 injections in neovascular AMD. In DME also, from drcr.net, we came to know that at least 13 to 15 injections are needed in a duration of 3 years to get a good result. So that imparts very big burden. In 2013 to 14, spending on specialty drug, including biologics, increased 32% in US, and small molecule drugs increased just 6.8%. So we can see why there is a huge burden of biologics. By 2016, 8 of 10 top selling drugs were biologics in US markets. In our country, why we face a challenge? Around 18 to 22% of Indian population is below poverty line. India's per capita income is very less as compared to developed country, and we are still a developing country. So it is a very big burden, and we cannot afford to spend that much on just health, and then all other sectors might be ignored. So the treatment costs are not covered. Many a times, if the treatment cost is very high, then insurance companies are reluctant to incorporate that reimbursement scheme into their system, or otherwise if they include, then the premium charges for the insurance goes very high. So again, the patient may not be affording that health insurance. So approximately 70% of our patients pay from their own pocket, and in underdeveloped part of the country like our part, that number or percentage is still very high. So there must be many dropout from the treatment, partial treatment, and then biosimilar here makes the medication affordable, help to reduce substantial cost burden to the patient. All the global coalition of regulator voices confidence in biosimilar, not only ophthalmology, from other all these rituximab, adalimumab, which we also rarely use in cases of resistant uveitic cases. So why biosimilar? Cost saving may translate into other associated benefits also. If the cost is going to reduce, it will have a impact on overall development of that industry or health industry as well. Reducing the cost of therapy will with increased compliance and success of treatment, a wider availability of treatment, and it will impact growth and development. And how it will impact the treatment regime also, there must be some newer development. Once that off-patent innovator is facing a challenge by biosimilar in terms of cost and market share, they are forced to work on newer molecule who are more efficacious, and they try to find out, and then we get new molecules which are more effective, and in that way the medical science grows. Many a times this availability of biosimilars at lower cost changes treatment preference as well. Like we remember when we were fellows, we used to do a lot of lasers to macular edema, even if center involving, and we see some of the microaneurysm and whatever, and if the patient is not affording anti-VEGF, okay, then you can undergo a laser treatment, focal laser, or you can take IVTA at the risk of development of cataract and raised intraocular pressure. But once these biosimilars have come, now we are almost switching to intravitreal injection of anti-VEGF for all kind of DME, and this laser and other things are just an adjunct treatment. So our treatment pattern also has changed with reducing cost burden of the treatment. Cost saving can be used to increase patient access to therapy also. Many a times if a patient is coming, he is very poor and there is no treatment option available, the patient is lost to follow-up, and even we are helpless to offer them any help in terms of treatment. So if the treatment cost is reduced, more and more patient can be benefited. Another thing, if a patient is developing some early mild edema, and if they are not affording, we would try to wait, okay, you just control your diabetes and we will see what is happening, and they may come in 3 to 6 months with a big edema. So but if the cost is lesser, we may try to start the treatment early, and then the results are good and the outcome becomes better. It has increased healthcare personnel as well. Reducing cost of the treatment has increased the number of healthcare professionals learning the technique of intravitreal injection, learning the technique of interpreting OCT scans, and learning the indications of the treatment. And now we have seen that majority of our friends who were practicing in lower strata cities, smaller towns, where they were not using anti-VEGFs or biologics as they were high cost treatment, and they were little bit hesitant that if I am going to inject a 50-60,000 injection, if the response is not good, then I may not be able to explain the patient. But once the treatment cost has come down, the confidence of patient as well as our colleagues have grown, and now the treatment has become widely available across the country. So that was the impact of reducing financial burden due to biosimilar. Cost effectiveness. The lower prices of biosimilar definitely improve cost effectiveness. Cost of therapy of individual and financial burden of government is also reduced, and that impact is transferred on the finance, growth and development of the country as well. Reimbursement, that is a very important thing. All third-party administrators are now willing to approve all this daycare treatment facility of intravitreal injections, because the drug cost has reduced, the procedure charge has reduced, so now they are including this reimbursement of injections in their scheme, and still that premium remains lower side, so more and more patient can afford through TPA. Even government is also now willing to accommodate all these costly injections because the cost has come down in their social welfare schemes like Ayushman Bharat. Now anti-VEGF is also approved in Ayushman Bharat, so very poor patients who can never afford on their own pocket also, they can get the benefit of this social welfare scheme. Just because the cost of drug has come down, even government can afford to offer to the poor class of the country. So this improves compliance and success of treatment. Thank you so much for your kind attention. Thank you, sir.
Thank you, Dr. Tiwari. So it was, very nice to highlight. And I especially took one point, which was quite striking, that we have been now seeing that a lot of people in the second tier city, third tier city, they want to learn anti-VEGF because previously it was a costly therapy and people were definitely, as he said, they were thinking that, if I counsel for an injection of 20-25,000, then if patient does not improve, then it impacts my practice. So that fear has come down, means that's what a great implication, which biosimilars are meant for, to increase the access of this therapy to more and more people. So that's great. I have one question, Dr. Tiwari, means you deal with a lot of patients, a lot of patients from surrounding areas from Patna, they come, how do you counsel about biosimilars?
See, whenever I'm explaining them, we have to give all the options because we never know which patient is going for what injection and what is their pocket just by the appearance. So we do explain that this is the injection which is being manufactured and marketed by international agency or international company. But this one is exactly the same molecule and it is being used for a longer time, and there is no difference between the two. The cost is lesser because it has been developed in India and it is being marketed in India. So the cost of therapy is lesser. In our experience also, we do not see any difference between these two. So whatever your budget allows, you can opt, but practically there is no difference, and these two molecules are approved also in India. So you can go for, and majority of our patients do opt for biosimilar, and we are also happy that we are giving them good treatment at lower cost.
Do you had ever had any questions coming back from a patient that can you tell me the difference, that with just India?
Yeah, they do ask because the cost of the treatment is around 8,000 to 10,000 extra in innovator and biosimilar. So they do almost always they ask that if these two are same, why the cost is more. I just say that this is imported from outside country, so definitely the cost is going to be higher. Otherwise, there is no difference.
Dr. Dogra, you have a different kind of practice in a different kind of setup. So do you get this challenge in terms of disbelief on biosimilars?
I think one of the additional thing which we always must explain once we put patients on anti-VEGF is the long-term treatment and multiple injections.
Yes, sir. Yes.
I think once we talk about that, and then they look at the entire cost and everything, and once you already said that the two drugs having similar efficacy and safety, which has been proven now. So I think that part is also very important. Once they calculate that, and we have most of the diseases where the drug would continue for a very long time, I think they get very well convinced. And that is another point we should keep in mind, and not tell them just 3 injection lagenge. No. That is a problem with many of our people, those who are not seriously into this. So then it is a problem. That's what.
So point means very well highlighted by Dr. Sir, because if we tell them from the beginning that this is the spectrum we are looking for a year, then probably that cost runs in the brain. Otherwise, if just we start treating in the beginning, then probably that math is little different. So means sir has a practice of a little more educated audience, means I would say educated patients. You have ever had any kind of patient who is saying means I have a little less belief on this cheap therapy?
Not really. I think it depends again on your counseling, which is very important, because since I came from absolutely a government institution where we had a different strata to something which after my superannuation to a practice which is a little different. But I think it's amazing. I'll show you some data also that even in an institute where I work, where many of the elite people come, they want Razumab because they know it that they have to spend even for so many years. So it's fine. I mean, a lot of our AMD patients, I would say, diabetic patients, because they require injections a lot. So they want this. So I think there's a big change,
Great.
I must say.
Thank you so much, sir. So I think now I would just rewind the clock and I'll invite Nilesh, because just to give us more understanding about biosimilars, that what exactly, because there are 2-3 terms which are being floated and highlighted, and sometimes it is different for a lot of people to differentiate these terms. Thank you, Dr. Tiwari. You can, thanks for all your help.
Thank you, sir. I would like to thank first Ashish sir for inviting me, and it's a privilege to be a part of this such a wonderful panel. So these are my disclosures. I am going to talk about biologics, biosimilars, and biobetters. What do they mean? So this is the public perception as what we were talking about. What is biosimilar? It is just a cheap copy of the innovator or the main thing. That's what most of the sad to say, because innovator companies also have created the conception when the biosimilar came in, not in ophthalmology, but in other fields. This was a gimmick that was played out. But then as soon as our understanding started, we got to know what it actually is. It actually holds the same basic structure. It has the same passion. It has the same primary structure. So all the amino acid sequence in innovator and biosimilars are exactly similar. It's just during the post-translational modification when the protein is formed, and then when it is getting folded, it's a secondary structure, tertiary structure, then the post-translational modification, if it is having a pegtail or something, there the difference lies. And that difference is not that much. It has the similar efficacy and similar safety. That is the baseline. So each drug, even if it is not having the same the quaternary structure, what we call, it has the same primary action site, and it has the same secondary action site, and it has the same efficacy and the same safety. So by definition, there is no standard definition across all of these medical agencies, but the crux what we could find was highly similar to approved biologics, notwithstanding the minor differences in clinically inactive components having similar safety, purity, and potency. So how are they manufactured? So the manufacturing actually...
Can I just interrupt, Nilesh? Go back to the previous slide. I can interrupt him. He was my student. So this is, a very important part that, you cannot be same. A biosimilar cannot be same as innovator. And that's where somewhere negative perception starts. That these are copy, they are not similar. But now it's important to understand, if you take a batch of Lucentis from Europe, if you take a batch of Lucentis from US, they would be different. Correct me, Alok, if I'm wrong. So because biologics are, kind of molecules which is very difficult that you really match each and everything. They are not generic, they are not chemical formula, because they go through the living organisms, and living organisms, there are few changes in terms of buffer, chemical are there. So that's what it defines, that clinically inactive component. So those are the components which do not matter in terms of the efficacy and the safety of that particular drug. So always remember, even two different Lucentis are also different. So it's not something that only biosimilars are different little bit, but they are all into the inactive component. So that negative perception should not come. Thank you. Sorry to interrupt.
No worries. So the workflow starts when the patent, actually it has to start when the patent expires, but as we know, R&D starts much earlier. So there is a reverse engineering on the proprietary molecule. So there is a lot of analytical, there is spectroscopy and more analytical thing that goes, so that they can find out what the actual primary amino acid sequence is, because that is a proprietary thing. The innovator is never going to tell, even if the patent expires, that doesn't come into public domain. So the basic amino sequence is interpreted and kept same. Then the secondary, tertiary, and quaternary structures, they might be different. So that the thing is to understand is the cell line from where the molecule is being created. It has to be similar. It has to be, if it is E. coli, it has to be E. coli. But exactly the same cell line basically is not used and should not be used. The cell line for production might be different, and the purification process might be different. And this is a point that we will ponder about when we are talking about betters, because the new technology, the biosimilars which are getting manufactured right now, are 20 years ahead of their innovators. There is more purification, there is more better cell lines that are being used to protect. So biobetters, when we come to, we will know why this is important. So then the workflow, Ashish sir will talk about. This is what I say. Let the master show how it is done, how they are regulated and how they are studied. So why do we need them? There is what Tivai sir was telling about. There is a direct discount that is happening, and then there is an indirect benefit from the lesser disease burden. So the patient who are getting treated, they are more economically active, and they are contributing more into the society. But what is inapparent is the wider availability playing to demand and supply. So when there is more supply, the demand, when there is more demand and supply is more, the prices come down. So that's what is happening. We are seeing with innovators, they are now they have started patient benefits, which was not available earlier. But as soon as the biosimilars started coming in, the patient benefit came, and even in the biosimilar, now the prices are coming down. And here, one point I want to tell about our pharma partners: please stop these schemes, 10 pe 2 free, 20 pe 5 free, because anyway you are going to give that amount to everybody. But if you are going to get the baseline below, so even if it is like 7,000, 8,000, 10,000, whatever the baseline you are going to create, if it is flat, the smaller practitioners who are going to inject one injection per month, they will also afford to buy that and inject that. So please stop these schemes and give a flat rate. So more and more people are going to use it. So this is a sincere request. And is it all rosy? Is there any side effect from biosimilar? You can see the graph that is about antibiotics. We are right now into that ascending phase. There is innovation that is going to happen, and then there is a plateau, and then now for last 30 years, we don't have a new antibiotic. We are in the ascending phase of this biologics. We are having every 2-3 years, we are going to have new molecules. But soon we are going to plateau, and there my humble request is don't stop innovation, because as you can see on the other chart, that is the generic chart, the amount of economics that is into play for this generic. So as more and more generics are being produced, the innovation keeps coming down. So as more and more biosimilar will keep coming, the innovation should not stop. That is one other sincere request from my side. So what are biobetter? Dhoni is the biobetter of Kapil Dev. Do you agree or not? There was already one World Cup at that time. Now he has gone ICC trophy. He has got T20. Similarly, some can say Kapil ke time mein there was no T20, there was no ICC Champions Trophy. Exactly the point. We have now better manufacturing capacity. We have now better purity. So that's why that is going to be a biobetter. It is but natural that they are going to be biobetters. Biobetters, there is no standard definition, but it has similar primary structure as biosimilar, but it has greater potency. Either it can have greater potency, it can have greater shelf life, it can have greater half-life. Anything can happen, but in one or more than one aspect, there will have a better, it will be better over the biosimilar. So these are the two potential very great molecules having very great efficacy, but they are marred. Abicipar-Pegol, it couldn't come into market because the IOI rate was at 8%, and brolucizumab, after being launched, is not being used that much because of the high IOI rate. So these in brolucizumab, it's very simple. You add a pegtail. You reduce the antigen component, like what we are injecting. If you reduce, there is high chance that the IOI rate will come down, because it's just what we propose. It is a type 3 reaction. The less antigen you inject, the less antibodies it will interact with and lesser IOI. So brolucizumab is one where biobetters will help. And abicipar because of the more purity that requires from E. coli components, so it will also benefit. But why there is no biobetter? Because it is considered to be a new drug. It won't have a abbreviated licensing protocol as biosimilar. So again, the entire R&D. So even if the initial cost of developing a molecule comes down, the trial cost is same, and that's why we need a regulatory change to get biosimilars. Each of the company, personal experience, Rani has no financial interest, but Rani Eyes has better drying capacity than the innovator molecule. Razumab also was better over the proprietary, but Rani Eyes, because it is more recent, it has even better drying capacity. But no company, neither Lupin will claim that, neither Intas will claim that, because for claiming, they need to go through that entire thing. So it is only whispers. So there is required a regulatory change to bring biobetters. Thank you.
So thank you, Nilesh. I mean, that was very well highlighted. So biobetter, I'll just put a point on biobetter that actually, if you really see in a simple sense to understand biobetter is, actually Lucentis was a biobetter of Avastin. Because Avastin was a full-length molecule with FC portion, with FAB arms, and what was done, that it was seen that, it works, and, so it was just modified in a way so that molecule size decreases, and you cut down FC portion. So ultimately, that's how the biobetter happens. But what happened? Lucentis came as a innovator molecule. It was essentially a biobetter. So biobetters means have a long road to, really have that regulatory hurdles. Means we understand a little bit. I think I would like Alok to comment on this. Means where, because he is been into industry for quite some time. So biobetter, what do you see, Alok? Means Alok is, he's Dr. Alok basically. He's from Intas. So he understands. Means I keep taking his guidance for many of our papers. So means he is the right person to comment on that.
I think biobetter is always an interesting thing, which is like somewhere you need a complete big innovation. Somewhere frugal innovation works. There will be no ways where we can see what is better or what is the way to follow. But I think even regulators now have defined a particular pathway for biobetters. And because of a gap in understanding how much innovation is real innovation, they've kept it more like a innovator product or a complete new product which is to be which you need to discover. Not probably from a biobetter example. And you shared a fantastic example. Lucentis is nothing but a biobetter of bevacizumab. We had certain histories with biobetter which probably didn't work when the numbers were expanded. So typically the trials, you see sometimes a very minor difference in a smaller trial, which when you replicate on a higher population size, that just disappears somewhere. And it's not always a statistical error. But just that, you include more people, that biobetter most of time becomes to be a biosimilar. So a classical example, when Dr. Nilesh shared the drying potential, absolutely we make a comment of drying potential on our observations, which are the newest first. But when we put it to the numbers, and I can vouch with all the trials, Rani Eyes trial is published, Razumab trial is published, you see CFT thickness same. So that's where biobetters run in trouble.
The trial are designed for non-inferiority. If we have a statistically superior trial design, this might be different.
So two things into it. They are designed for non-inferiority to reduce the population size involved. Rest, non-inferiority and superiority trials, if you design, typically you are only the subject size changes. Apart, of course, from your primary endpoints and statistical plan. But that's where that biobetters doesn't have yet been established as a fact, because you never know if you put it to the mass, which way that biobetter would work. Actually as an innovator, or that small difference seen might actually become nullified in the efficacy or safety
when we see. And that's where the problem is with biobetters.
True, true. So means, but I'm glad to hear that some path is being given now. So Dr. Vidya is from Lupin. I think what I have heard from them is means they have, because they are the, I think, latest candidate here, and then their endotoxin limits are very, very stringent. So means I want your comment on that on the same question, that biobetter pathway. You had some comparison of the endotoxins as well, isn't it?
Yes, the data is not valid, not publicly enough. The data is volume. So when we have compared the other biosimilars available versus 2 lots of different batches that we have manufactured in our in-house facility, we have come across the data like more or less all the biosimilars are similar in terms of, say, relative binding potency and the other characteristics, the critical quality attributes. But yes, as Dr. Sharma has pointed rightly, the endotoxin limits, it's something we are more worried about because it directly impacts the efficacy or safety of the molecule more than efficacy. So yes, we have found that the endotoxin limits are very much below than the accepted criteria what has been set by the regulatory bodies. And I think that is the reason why we are not seeing much IOIs, much complications right now in last few years.
Much, are you seeing it?
No, so far, touch wood, we have not come across any case. We have sold.
Okay, so means that's what happens once you keep getting better and better. Means, as Nilesh also said, these are subtle things which could make a molecule better, but you can't really put it, as Alok said, in a perspective in terms of to say it. Yes, thank you so much, Nilesh. Now I would invite most respected Dr. Mangad Dogra sir to give and share his experience. I think he has been teaching us on various aspects since childhood, that when we were all children in this field. Thank you so much, sir.
Thank you, Dr. Ashish, for giving me this opportunity to be part of this very, very important area, which is evolving in a big way, which has been talked of. So I'm just going to give you, I think I must say here that I have some financial disclosures here where I do at this stage now as a speaker for them, or sometime attend their meetings. I think once we talk of biosimilars in the country, what we started with Razumab, and then Retinal, and now Ranizuril. So I think these are the three. Of course, some companies are sort of sharing the product. That is what is happening. And one thing is there, that as far as the anti-VEGFs are concerned, only Ranibizumab biosimilars are approved at this time. I think there's a lot of work going on Aflibercept biosimilars. So there are going to be a lot of competitions in future. So it's going to happen, but they still have a patent restriction. And so I think if we look at various trials which have been done, especially for drug approval, they have almost been similar. Maybe patient numbers are different, but that's how probably the approval was possible from DCGI for all the three in this country. And as far as the real world data is concerned, I think it was not 7 years, now it is 8 years now as far as the Intaz is concerned. And it was right in the beginning, 2015, approved for almost all indication, except the additional approval was for ROP subsequently. I still remember, I think Manish is here, he has turned grey, when he would visit us in PGI and we would not use it. You see, that is what happens, and that is a hesitance. And of course, we have the same hesitancy with the later two, Ranizuril and Reniais. We want more people to use it and then become confident. I tell all the time whenever the people are visiting us. And I must say, as far as this ROP indication is concerned, this Janani scheme by Government of India, you see now these days everybody is injecting Razumab in ROPIs. And that is itself, data should be shown from this country. And I'm sure the data would come. And I think as far as ranitidine is concerned, I have already shown this, that they have shown safety as well as efficacy. The total injection used maybe around 15,000 or maybe a little more than that now. And also for the lupin, this was maybe the numbers have gone beyond 15,000. And do we have most of the data? I think it comes from this country only, and mostly where Dr. Ashish and his team has been associated, as well as some of the other data which we saw already today from Debulal. I think we need to be kind of going into that kind of exercise where we generate data, we have evidence. That is what is very important, and that's how it will become a global phenomena. And I think this one particularly here talks about, again by Ashish and group, where they talk of ophthalmic biosimilars from India, and this was in 2019, and a lot of things have, and even VRSI has done a survey, and what that survey revealed, that Indian vitreal specialists are familiar with anti-VEGF biosimilars, and there has been now progressive trend, like what I will mention and show to you, that we are accepting and using biosimilars over biosimilars, and there is a need for enhanced pharmacovigilance. I think VRSI did have that kind of adverse, still they have adverse event kind of a site where they look at, so that part is very important always. I think basically, the Razibem, when it was launched, 45% cut, then the Lucentis, then price cut has gone up to 66%, and others also like almost similar. And the prices of the innovator, because of the same reason, they had to bring down. There was no other way. They would have probably otherwise gone out of the... This is just showing, this is, my son is now consultant in PGI. I just asked him, just give me rough data from what you do with this. So you can see here, this is a big institute where I've worked all my life. So out of the like 40, which he uses himself, they have a large team. There's so many of them. There may be more than 10 consultants. So 20 are Razumab, 10 eccentrics, 5 to 6 Eylea, 3 to 4 Ozurdex. So why I'm showing this, this is where the place, I think Manish couldn't come, and all indications, you can see all indications are shown there. You know, they entered with great difficulty in October 2021. I superannuated in 2019, and that is the Garewala Institute. And so I asked my people, just to take out the data. So Razumab has gone up. That is the number one. So it 981 since then. And you can see eccentrics and Eylea because of the scheme. Ozurdex and Paginex is really low there. I think we're still not using. I just wanted to show, look at this 82 years gentleman, how well he did with 3 injections. So that is seen here, and the vision improved. You see, that's how the confidence is built. And this patient, since this is something where he's paying out of the pocket, he is going to keep on getting the, and that is what he's doing, injections. This gentleman, this was with me from PGI days. You see, every time this wound never dry, and because he was not receiving enough injections. So it is 660, 624. It was something I told him. Okay, we have this new drug, which is cheaper, and I'm going to give you 6 injections. And it's unbelievable. You can't. He's the happiest man today. After 6 injections, both eyes become dry. Look at his vision. Now, his near vision has also improved. And somehow, now his requirement is every 4 months or so. I mean, which is unbelievable. So I think things are happening, and vascular occlusion, we're going to listen from our keynote speaker here, and just within 1 injection, it shows beautiful, you see, resolution. So in summary, from a very slow start, which I mentioned already, and I think today the confidence has been built, and we had a problem, and this problem was that inflammation which happened, and it happened a couple of times, and we won't use it. And once we have large number of patients, they have already more than now 5 lakhs already done in this country, the numbers are going to increase. And I liked the suggestion by Nilesh, if they kind of incorporate that thing into the main market, more user would definitely be there. And safety is established. And there's no doubt that biosimilars are the future. And that has happened with other drugs in our country. How to adhere to the treatment. The cost is very important where people pay out of the pocket. So I think that is where we are. But if we have more data, it will become a worldwide phenomenon, I'm sure. Thank you.
Thank you so much, Dr. Dogra, for highlighting very well facts, which means that Grewal Institute data and from data from PGI was just excellent. You see, means from where it started in 2016 with so much of skepticism. And that's where US stands at this point, I would say. Means yesterday itself, I had a meeting with Biogen, and it still means that acceptance is not there. So that's how we started. And that's how, if you look at the VRSI survey data, it's just climbing up. And you can take the example of filgrastim and any other biosimilar. It just slowly creeps in. And NHS, if you see now, UK, the policy is very well defined now. I'm not sure, sir, whether you're aware or not. NHS placed it in their policy that as a step therapy, you have to use biosimilar first in many of their provinces, because it goes through the provinces. So that's how it happens. So we are looking forward for aflibercept biosimilars that is going to transform the entire care, especially in this country, because globally, if you see, aflibercept is something which has the largest market share, but we are not able to inject. Thanks to their scheme now, as Dr. Dogra sir keeps saying, after that scheme, my consumption has gone up, patients' adherence have improved. Again, that shows the financial aspect. So we have talked a lot about India, but I would now touch upon one point which Nicola will talk about on-label bevacizumab, which is a very, very hot topic in America at this point. American system is completely shaken up by this. They are very, very skeptical. I'm not sure whether you have read a recent paper on ophthalmology or not, published a couple of weeks before. I have written a letter to editor already. So that paper talks about that if on-label bevacizumab gets approval by FDA, the heading was that on-label bevacizumab, if approves by FDA, overall biosimilar are going to increase the healthcare cost. But everything was hypothetical. Biosimilar on-label bevacizumab price was hypothetical. They have not mentioned that, what already approved molecules. You have two biosimilar ranibizumab already approved there. What they have done, already 40% price cut is there. But US is somehow a little complicated system in terms of the insurance, that how much they pass on. So she's going to talk about that. It is just there. If this gets approval, a lot of things will shake in United States especially.
A very good afternoon to everyone. I'll be talking about on-label bevacizumab. I have no disclosures. And before I start, I would like to thank Dr. ASHISH Sharma sir for giving me this wonderful opportunity to be a part of this instruction course. So the drug bevacizumab or Avastin was first FDA approved for the treatment of metastatic colorectal carcinoma. And this was at a time when the only treatment for AMD was just PDT, and ranibizumab was just in the final stages of approval. It was Dr. Phil Rosenfeld who pioneered the use of this intravenous agent as an intravitreal agent for the treatment of wet AMD. And this was in 2005. But a big problem in using this intravenous formulation was that it had to be compounded or aliquoted. So unapproved repackaged bevacizumab provided by these compounding pharmacies had the risk of contamination, inconsistent potency, and availability. Another recent problem was the expiry of Avastin's patency. So because of this, 2 new biosimilars were approved, and this was in the field of oncology. The first one was bevacizumab AWWB, and the recent one was Zirabev, which is bevacizumab BVZR. So ophthalmologists were not very keen on using a biosimilar of a drug which was not approved in the field of ophthalmology. So then came Outlook Therapeutics.
So can I interrupt you, Nicola, just for a second? Just go over the last slide. So what happened in US around 6 months or 9 months before, there was a short supply of Avastin. Because still, Avastin has a lot of belief in US system, because that drug is cheaper than what we charge for Avastin in India. Around 50 to 100 US dollar. That's what the price is in United States for Avastin. So what happened? There was short supply from the company. So in between, there were two biosimilars which were approved, Zirabab and another biosimilar. So they were pushed by the insurance companies. But because Avastin has plethora of evidential data, clinical data, so it was quite, resistance was encountered from all the retina specialists. I mean, that's what she is trying to highlight. So because that's where, the clinical data, importance of clinical data comes in. Please, sorry for interruption.
So Outlook Therapeutics has come up with the drug, an on-label bevacizumab, which is Litinava ONS5010. It's a full-length humanized anti-VEGF and it is an investigational ophthalmic formulation. Basically, it will be an on-label bevacizumab approved for wet AMD if the approval happens. So the big question, is this drug a biosimilar or an originator? The drug Litinava, bevacizumab VIKG, is not a biosimilar. It is an innovator. Because for a product to be called as a biosimilar, it has to have a reference product or an originator that has been approved for that specific indication in that specific field. So Litinava was never had an originator product which was approved for wet AMD in ophthalmology. Bevacizumab was only approved in the field of oncology. So Litinava will be an innovator. And if the approval happens, it will be available. It is expected to be available both as vials and as single-use syringes. The drug at present has completed 3 clinical trials, namely NORSE-1, NORSE-2 and NORSE-3. So NORSE-1 is a clinical experience trial which happened across various centers in Australia, and this trial compares the drug ONS5010 with an alternate dosing regimen of ranibizumab. Since the dosing regimen was not similar, meaning an apple-to-apple comparison did not happen, a monthly to monthly did not happen, a monthly regimen with an alternate dosing regimen of Lucentis was compared. So this trial design was questioned. And then came NORSE-2, which was a pivotal phase 3 trial where 228 wet AMD patients at 39 clinical sites in the United States were evaluated. The trial design was a superiority trial comparing the safety and efficacy of the drug, which was dosed against the monthly ranibizumab according to the prior regimen. The primary endpoint was at 11 months where the 15-letter gain was met. Another scenario which tells us that the drug is an originator and not a biosimilar is that for biosimilars to get FDA approval, equivalence trials are enough and a superiority trial is not needed. And moreover, the drug ONS5010 was only compared with the FDA-approved drug ranibizumab and not with the off-label bevacizumab. NORSE-3 was a safety study. There were no incidences of vasculitis, 1 case of iritis. The frequency and incidence of adverse events overall were low, and the most common adverse event was subconjunctival hemorrhage, which was attributed to the injection and not to the drug. 2 more trials, namely NORSE-4, a trial that will evaluate the drug for BRVO, and NORSE-5 and 6, which will evaluate Litinava for the treatment of DME. Another scenario which tells us that the drug is a originator and not a biosimilar, because for a biosimilar to get approval, one indication for one clinical trial is enough, and from that clinical trial, extrapolation can be done for other indications. But for a originator drug, separate clinical trials have to be conducted for separate indications, meaning extrapolation cannot be done. So the US FDA at present has accepted the Outlook Therapeutics Biologics License application filing for review. And if the BLA is approved, it is expected to result in a 12-year market exclusivity for ONS5010 as the first and only ophthalmic formulation of bevacizumab approved by FDA to treat wet AMD. So what will be the deal breaker for this drug? Will it be competing with the newer biologics like faricimab for the longer treatment interval, or will it be competing with newer ranibizumab biosimilars in terms of pricing? So in the Western countries, according to their law, it is not justifiable to compound an off-label Avastin when an FDA-approved on-label drug is available. And moreover, the pricing is in such a way that the off-label drug is priced at around $75 US, and the on-label Avastin, if approved, is expected to be priced at around $750 to $1,000 US. So this strategy is in such a way that the price is low enough to move these off-label Avastin users to the branded Litinava, but still lower than biosimilars to make sure that these physicians will prefer to choose Litinava over the biosimilars. But in India, where in a developing country where pricing rules everything, the newer ranibizumab biosimilars, 3 drugs have been approved by the Drug Controller General of India. They are very economically priced. So a lot of insights for this can be gained from the drug Lumiere, which is an ophthalmic bevacizumab approved in Argentina. So in spite of being an on-label drug, physicians and patients still preferred the off-label Avastin because of the significantly higher cost of Lumiere. So to conclude, for this drug Litinava to be a success in the Indian market, it has to be available as single-use syringes to combat the problem of compounding, and it has to be economically priced, keeping in mind the newer ranibizumab biosimilars. Thank you.
Thank you, Nicholas, so very much. So, means important point to highlight is what US law implies is in case they have any on-label bevacizumab for ophthalmic use, which they never had, if that is there, then as per the law, they cannot use Avastin anymore. That's where the problem lies. Means, Alok, do you see any of the industry member, do you really see any future of that drug in India? Means, I do not see that, the kind of, biosimilar development that we have in this country. I had this specific question to you because you had one, biosimilar of Avastin in your systemic usage.
Biosimilar of Avastin came much before ranibizumab, and most of biosimilar companies do have Avastin biosimilar already active in market. The issue is not with availability in India or how it would be dispensed in India. Today, even if I look at Avastin, my price per dose of a drug is so low that I cannot go further down, and the gap because of the pricing. So typically, old time Lucentis 45,000 per vial. Avastin that time was also at similar price, but that gap has been drastically narrowed with biosimilars. And now, as the years have passed, the basic economy has come up. The need gap for reduced pricing has come down, and that's why if you see even the Outlook Therapeutics have not priced it, or in fact they are more similar to biosimilars pricing. So they are saying it will be around 40% discount to the
Biosimilar.
Lucentis,
yeah.
and biosimilars are already at 40-50% discount. So that's where the gap in US will also occur. Though US is quite enthusiastic about Avastin biosimilar, India, I frankly don't see a case because the moment I go into single packing, my cost of drug basic goes.
Yeah, that's where the challenge comes. That packing.
Which I will down, and again on top of it, I have to change my basic. I would say basic requirements like the small ones, excipients.
True.
Which I need to add, which makes
it a little more complicated. Thank you, Alok.
So now Servil is going to talk about the how do we understand about the regulatory trials across the globe, because each of the country has a basic structure, but there are certain subtle differences that he's going to highlight.
So thank you once again for each and everyone for contributing to this IC, and actually speaking, a lot of things which are there in the slide, they are already spoken. So thanks a lot for that. So before I proceed further, these are my disclosures. I'm going to talk about the regulatory. Regulatory is something which is kind of a pivotal point for biosimilars across the globe, because that's where a lot of nitty-gritty needs to, you need to understand. And even it took a lot of time for regulatory to come into streamline in various countries. And still they are not a streamline, means nobody's on the same platform even now. So we'll talk about that. So I would sincerely appreciate the effort of Nilesh and Nicola. This is our publication. This is a list of our publications. We try to understand on various aspects of biosimilars from various angles. And one of our paper we are truly humbled to say that has become the policy document for biosimilars in Canada. So, means thanks to this entire group of people across the globe who have been a part of all these papers and contributed in terms of reviewing and helping us to understand biosimilars much more. So regulatory approval process, Nilesh has already shown this, kind of pyramid. If you really look at this pyramid, that why do we have this pyramid? Whenever a new molecule comes in, you have no idea that how it's going to behave. So that's the reason that you do a large database of clinical studies, which comes on the base of the pyramid, has the highest volume. And you do those studies in RVO, in DME, in different indications, because you have no idea that how they're going to behave. So that's the reason that you have larger clinical trial database to be generated before approval as per the regulatory, which is right. But now with biosimilars, what happens? You see these clinical studies, they go on the apex. On apex, you have the limited amount of clinical data. Why? That's where the nocebo perception comes in for many people. But they should not be, skeptical about it, because you are comparing and you are trying to show it similar to a molecule which already has a track record of safety and efficacy. So what exactly you need to show and prove based on the pharmacology analysis is your primary structure is in place, secondary structure is in place, tertiary structure is in place, or quaternary structure is in place. And then still, if you want to understand and if you want to really, check it that whether some difference comes, that is done with the help of limited clinical data. This is the global regulatory requirement, not only India, across the globe. So this is the paper which I was talking about. Now, it is country specific, based on the requirement of that particular country. For example, if you look at India, these are the three major biosimilars which were approved, but just look at the clinical trial design. 3 months, 6 months, 3 months. Resumab started with the least number of sample size. Probably, they give a platform, to understand, how things were done. And at this point, if you look at the overall clinical trial design, probably Retinal had the best clinical trial design, almost close to, what global clinical trial requirements are. But, this takes a time. Overall, in last 7-8 years, understanding has developed. Patients were minimal if you compare with the global designs. Primary endpoints, if you see, they are approximately at 12 weeks, as I said during Dr. Debulal talk, that why it is so. Now look at the global biosimilars. You have patient number much higher, but still you see the patient numbers are not defined and equal. So even FDA is not defining that, I need this certain number to be tested. So they also differ. But if you look at the primary outcomes, almost similar. We differ in terms of loss, and here it is the gain of letters with the margin of letters, that how much you gain. Study duration is equal, 12 months. So as I said in Dr. Debdulal's talk also, it is just the efficacy which is tested at these short time points, because these are all bioequivalence trial. Bioequivalence, if you want to prove, you have to have short time points. Otherwise, as I said, if you go on 12 months, 9 months, you cannot prove that there is a bioequivalence was there. So you should not have that negative perception. This was the editorial, actually, it was asked by the FDA reviewer. When we were writing a lot of papers, then, he wrote to us, it was rejected once. Then he written personally to us that, Ashish, I want to bring that. I do not know how it got rejected, but it was again published just to avoid nocebo perception, which keeps on, prevailing on biosimilars, both in United States especially, because Europe is a little more mature in terms of the biosimilars. Now, as I showed the clinical trial data of India and US, and you must have seen total duration 3 months, 6 months, 3 months in India. But you have seen 12 months, 12 months, 12 months in the United States. Does this make Indian molecules inferior? It does not. Why is that? It's all about the regulatory requirement. Which regulatory asking what? So now if you look at the Lupin's global trial, they have the sample size as same as what FDA and EMA approved drugs are. Similarly, if you look at the even Resumab trial for their global approval for FDA and EMA, their numbers are as same as which were there for the FDA by the other molecules. So it's all about, which regulatory you are applying for. Probably Indian regulatory, the need is much, much more for this country. We are catering 1/6 of the population of this world. So probably, fast track designs are there in DCGI, so that's fine. This is different in even Japan as well. Japan did not do for 12 months. Japan sample size was less. So it's quite country specific. This slide was shown. So this slide means why I am showing this slide, because there again a regulatory involved here. What happened in 2015, initial 2 batches had some kind of inflammation. But over the time, you do not see that because regulatory changed. But again, the question comes, because we saw some kind of inflammation, does that make Indian molecule inferior? No. If you look at the Lucentis data, when it was in phase 3 trial, you had inflammation in anchor median of 17.1 to 20.9%. Can't you believe it? But you do not see that now. Because what happened over the time, the requirement by the regulatory, as I was discussing with Vidya again, I asked her to raise that point on endotoxin. Endotoxin limits have been strengthened over the time. That's the reason that you do not see that kind of inflammation with these molecules. And again, one trial approval, as Nicola said, that one trial approval, it gives to all the approval. Again, this is a negative perception, but again, a regulatory requirement. Regulatory understands there's a wisdom behind it, because Lucentis has never shown differential safety issues with different indications. That's the reason that even with biosimilars, you have one approval, same receptor, you have approval for all the indications. If Lucentis would have shown differential kind of inflammation, definitely regulatory would have asked them. Future possibility means this, I'm telling aloud. I see this happening after 10 years. But probably not today, means we were writing a paper on that, but my mentor, Dr. Cooperman, said, Ashish, stop at this point, because US is just starting to believe biosimilars. If you write this, the kind of belief that they're developing, that might go off. But I see that happening over the time, because UK has already slashed off phase 3 trial in many of their biosimilars, and there is a reason. You have inflammation percentage is approximately less than 1%. If you really scientifically see, to detect 1%, less than 1% of inflammation, the sample size has to go really, really wide, which you really you can't do that. So rather than save money, because the main advantage of biosimilars is saving money. You see the testament from Europe, there is no biosimilars has been withdrawn from the market till now. They are the most experienced nation. So education, which even FDA EMA is trying to put in. So novel asset like brolucizumab might give you surprise, but not the biosimilars, which really emulate the product, for which safety and efficacy is already established. So if we believe them, I feel that many lives can be better, not only in India, across the globe. Thank you for your kind attention. And now I would like to invite our keynote speaker, Dr. Jay, and he would enlighten us on RVO. We have time for your talk, sir. And thank you so much for being here. I think you were the first one to be here on this session.
Thank you so much, Dr. ASHISH Sharma, for your kind words, and thank you to Dr. Biswas, Partha Biswas, for his kind invitation as well. Although I was given a very generic talk of retinal vein occlusion, I thought I would modify my talk to fit in with the flavor of what's been discussed now. Is basically cost-benefit analysis, and it's not just developing countries like India, but cost is something that affects every country. As Dr. Sharma has rightly pointed out, our first line management now is Ongavia. So the difficulty we have is of switching patients who have previously been on Lucentis to Ongavia. Whereas it's not difficult starting a patient on Ongavia. If somebody is stable on Lucentis, it's more difficult to justify switching that patient onto Ongavia.
So sorry to interrupt, Ongavia is a different name, but it's the same molecule FYB201. Means BioVis, which was a molecule from Samsung Biopsys, that has the same name across the globe, BioVis. But Ongavia that sir is talking about is the molecule FYB201, which has different name in different countries, and Ongavia is the one which they call it in UK. Sir is from UK.
Yes, I mean there are 3 biosimilars that are available, and like most drug manufacturers, different names are given in different countries. So yes, it is a ranibizumab biosimilar. So I'm not going to talk much in general about retinal vein occlusion, because I think this is something that you all will perhaps all know about. It's the second most common cause of vascular-related visual loss, retinal vascular-related visual loss, apart from second to diabetic retinopathy. I'm not including AMD specifically in that. And there are broadly 2 categories, perhaps 3 if you include hemiretinal vein occlusion, but basically they are branch retinal vein occlusion and central retinal vein occlusion. And here we're specifically talking about the macular edema associated with retinal vein occlusion to fit in with the flavor of the talks that we have had previously. So fluid leakage in the retinal vessels is not just a mechanical effect. It's not like a hose pipe you clamp at one end and the pressure of water makes fluid to leak out from little holes in the tube. But it's a very complex underlying vascular and inflammatory pathologies that are involved. And we've seen that there's upregulation of vascular endothelial growth factor and various cytokines in retinal vein occlusion. We've been discussing that the management basically consists of intravitreal injections. And we basically have a choice of 2 broad categories of intravitreal injections. Either an anti-VEGF agent, and here I'm talking only about the NICE approved, NICE's National Institute of Clinical Excellence, which is an approval body in the UK. So either ranibizumab or aflibercept, and that includes ranibizumab biosimilars as well, or the steroid dexamethasone. So in terms of cost-benefit analysis, how do you choose what to use as first line? Do you want to try anti-VEGF and see whether that works? And if the anti-VEGF, after 3 injections, you get absolutely no clinical benefit or OCT benefit, or you get partial benefit and you want to try maybe 6 injections before you switch? Or do we switch early on? Or do we start with a steroid? And that is the question. In all situations, one thing to think about is the drug cost. But perhaps a larger proportion of money goes towards the service cost. That is, paying for my time, paying for the nurse's time, paying for the receptionist's time, paying for the person who pulls out the notes. So that is the overall cost. So by decreasing the number of visits, we can perhaps decrease the overall cost burden of treatment. So it's not just the drug cost that one needs to think about. So can pre-treatment assessment of aqueous biomarkers help guide management of these patients. And this was financed by, it's called the P2P grant, a University Health Board grant. The aim was to measure the level of vascular endothelial growth factor and inflammatory cytokine IL-8 in the aqueous humor of human controls, branch retinal vein occlusion and central retinal vein occlusion. And we compared the levels of these biomarkers, compared branch vein occlusion versus central retinal vein occlusion, to see whether this really helped us with a choice of treatment. So it was a very, very small pilot study of 30 patients where 100 to 200 microliters of aqueous was collected from the anterior chamber. There were 10 patients who were undergoing routine cataract surgery in those patients who had no retinal vascular disease, no view. So it excluded uveitic cataracts. It excluded cataracts in patients who have had treatment for AMD or diabetic retinopathy or retinal vein occlusion. So basically, squeaky clean cataract. So we felt that that could be taken as a baseline. And 10 patients each of branch retinal vein occlusion and central retinal vein occlusion. The aqueous samples were frozen immediately at minus 80 degrees centigrade until they were analyzed. And we used the ELISA technique on the Luminix MacPix analyzer using the double sandwich technique of indirect immunoassay. In terms of the vascular endothelial growth factor, because it was a small sample size and we found that the values were quite variable. Looking back into previous studies, there were a large variation in what various studies considered as controls. And therefore, rather than taking the mean, which can sometimes give a very skewed perception because you might have one very large outlying value, we thought we'd take the median as a better marker of the true value. So we found that these values are in picograms per milliliter. Control 62, central retinal vein occlusion 141, and branch retinal vein occlusion 85. So the difference between the controls, which is cataract patients, and central retinal vein occlusion was statistically significant. However, there was no statistical significance between the controls and branch retinal vein occlusion. Likewise, when we analyzed branch retinal vein occlusion and central retinal vein occlusions, the VEGF values were much higher, statistically higher in central retinal vein occlusion as compared to branch retinal vein occlusion. This is just a different way of depicting the same data where you see the central retinal vein occlusion really had much higher values than the control and branch retinal vein occlusion. In terms of interleukin-8, the control values were very small. Central retinal vein occlusion was much higher. And in branch retinal vein occlusion, it was higher, but not as high as central retinal vein occlusion. But both the central retinal vein occlusion and branch retinal vein occlusion, the levels were statistically significant. And likewise, comparing branch retinal vein occlusion to central retinal vein occlusion, and that difference was also statistically significant. And this is just a chart to show what we found. But what we found interesting is that individual values were highly variable. There were some samples where VEGF was the predominant marker. There are other values, other samples where it was not the VEGF, but the cytokines, inflammatory cytokines that were that were the major marker. And the same thing was true for central retinal vein occlusion and for branched retinal vein occlusion.
So just a kind request for the
Sorry?
quick conclusion.
Quick conclusion. So
thank
the
you
basic
so much.
conclusion was the level of VEGF was higher in central retinal vein occlusion as controls, and likewise in branched retinal vein occlusion. So the question is whether we can use these biomarkers to decide which drug to use. We, of course, have had small sample sizes. Perhaps what we need to do is to increase the number of sample sizes, increase the range of biomarkers. We've taken IL-8, but there are now kits that are available that can simultaneously measure up to 27 biomarkers for the same sample size within the constraints of very, very small samples. So in short, maybe if we tested these biomarkers, we can go in directly for a drug which is perhaps the eye might respond better to. Thank you.
Thank you so much, Dr. Jay. I have questions, but I think in respect of next session, I will, off the podium, I'll ask those questions and discuss with you. Thank you so much. Thank you, Dr. Mangat, and thanks each and everyone to be here, and thanks a lot.